Okay, why don't we go ahead and make a start on today's SimSip webinar. So welcome everybody. Just a few housekeeping notes. All attendees are out and muted, but you do have the opportunity to ask questions. To do so, simply raise your hand and unmute, or alternately, you can also type your question in the chat box, and we'll ask those questions at the end to Trevor at the end of this presentation. We are recording today's presentation, so it will be available on YouTube following the presentation should you want to review it afterwards, or please share it with your colleagues who were unable to make the live session. So with that, I'd like to move on to introduce today's speaker, Trevor Johnson. Trevor, would you advance the slide? Thank you. So Trevor Johnson is a principal scientist at Certara UK's limited SimSip division. As you can see from Trevor's prior work, he's a former pediatric hospital pharmacist, and he also received his PhD in pediatric clinical pharmacology. All that to say that within the pediatric space, Trevor has a viewpoint of a clinician as well as a researcher. He has extensive experience in pediatric clinical pharmacology and brings that experience to develop PBPK models for pediatrics. But not only that, he's also worked with other special populations such as renal and hepatic disease. And if you have an interest in pediatric clinical pharmacology, it's highly likely that you have come across some of his work with over 100 research publications that Trevor has been involved with, which have developed specifically on developmental pharmacology. And so leading today's talk, Trevor will be discussing optimizing the CYP3A4 ontogeny in pediatric PBPK models, specific looking at hepatic 3A4 ontogeny and intestinal 3A4 ontogeny. And he'll also be discussing using available pediatric clinical data for model verification and some of the issues with that, and also talk about possible future research. So with that, I am super happy to turn it over to Trevor, who will be covering the subject I just mentioned. Trevor? Trevor, you might be muted.
Thank you, Jean. Can you hear me okay now?
Yes. Yes.
Right. Thank you for the introduction, Jean. As Jean's already said, I'm going to be talking today about almost like a lifelong research theme that I've had in terms of looking at the ontogeny of enzymes and processes in pediatrics, and specifically for pediatric PBPK models. And I'm sort of tongue in cheek asking, are we there yet? I think, you can decide at the end of this session whether we are there yet. I think we're getting there, but we're not there yet, is my sort of early comment on that, really. So I'm just going to give you some background, really, where the ontogeny data originally came from and how it's really evolving over time. And then I want to look at how we've tried to optimize the hepatic CYP3A4 ontogeny, and then more recently had a look at once we thought we'd got the hepatic 3A4 ontogeny correct. Then went on to look at the intestinal 3A4 ontogeny, which, as you'll see later, is still quite an active area of research, really. I also want to sort of explore a little bit about, the available pediatric clinical data for model verification. And what you've got to often remember about a lot of the clinical data that is there in the literature, when people are generating that data, they're not really thinking about verifying PBPK models. They're mainly clinical studies, and they've got some sort of clinical question that they're trying to ask. But I think there is issues on how some of the clinical studies are not necessarily undertaken, but how the results are presented at the end and whether that could be improved, both to improve the sort of clinical information that you can get and also help us to verify some of the models that we're interested in. I'll briefly go at the end, just go into some future research as well. So the original CYP3A4 models that we put into the early versions of SimSip Paediatric were really based on in vitro ontogeny data. And all I've done on this screen is highlight a few studies. So the original paper I wrote in 2006 was really a pre-SimSip version of a pediatric PBPK model, and the data came from these top 3 publications here: Lacroix, Jeffrey Stevens, and Treolia as well. So there's been a fair bit of interest by some of the French research groups in this early ontogeny work. And it really sort of started, in the sort of 1990s as the CYP3A4 homology had been sort of worked out, and they got it down to specific isozymes where they could look for the ontogeny. You do see earlier studies than that, but it's more generalized and often involves, what was recognized at the time as a probe substrate, and what subsequently may be an accurate probe substrate, or maybe a sort of not very accurate probe substrate for a specific enzyme. So these top three publications were the original ones we put into that paper in 2006. And all these publications on here are really the ontogenies that we then put into Simcyp version 13. What you'll see from the next slide is actually they're not really that much different. So, we had originally a lot of in vitro ontogeny data. And in general, it was telling you the same thing, that, early on there was low levels of CYP3A4, and as the child aged, it developed to get to an adult expression roughly around 5 years old at the time. So this was fine. This was a good starting point for us, really, in terms of incorporating ontogeny into Simcyp. Then you always get work coming along that makes you doubt that some of this is correct. And this study by Leong et al., this was from really from FDA people. They put this paper out, it's 2012, I think, looking at regulatory experience with PBPK modeling for pediatric drug trials. And they used Simcyp as part of this evaluation. And what they did, the graphs are probably fairly small for you to see, but they looked at three substrates: midazolam for 3A4, theophylline for CYP1A2, and omeprazole for 2C19. And what they generally showed there was that actually Simcyp version 10 compared to the observed wasn't doing that well in terms of predicting clearance. And you can see from midazolam, theophylline, and omeprazole, we were basically under predicting clearance for all three drugs. So that was the general picture, the under prediction. So the possible reasons were, maybe we've got the ontogeny models wrong, maybe they were incorrect, and we need to sort of modify those. But before we did that, we also looked at other things as well, like milligrams protein per gram liver. Was that scalar correct? Was the protein binding correct, etc.? And when we look round at the different parameters, with the possible exception of protein binding for certain drugs, but not these drugs, we thought that the other parameters were relatively robust, and actually work since then, especially on MPPGL, has shown that how we think that change with age is correct. The other thing with this analysis, there are certain problems with it, and I'll not go into all of them, but if you look at the midazolam graph on the left-hand side here, you can see actually they didn't predict that well in adults. And we always say one of the rules for pediatric PBPK predictions is get the model working in adults accurately before you go into pediatrics. If there's anything wrong with your drug file, you need to sort it out in sort of more in health with healthy or relatively healthy adult studies before you go into pediatrics. There were other issues as well. There were a couple of studies in here where I think one of the midazolam studies, the 12 to 16-year-old was N equals 2. So it was a tiny, 2 subjects aren't representative of a population. One of the other interesting points was that for some of the omeprazole studies, there was massive variability in the data. So if the average was falsely high, and if you actually looked at geometric mean predictions, we actually did okay there. But I think overall, what these studies highlighted to us was that maybe we ought to look at some of the ontogenies again, and we went on then as a group, and Fazana Salam led the project as part of her PhD to reevaluate and validate ontogeny functions for CYP1A2 and 3A4 based on in vivo data. And just after her publication, a couple of years after, there was another publication by Vijay Upreti, who was based at Amgen at the time, and they did a sort of similar study to this. And interestingly, if you look at both studies, they more or less agree on the CYP1A2, revised CYP1A2 ontogeny, But if you look at the graph on this slide, you can see actually we disagreed on the CYP3A4 ontogeny. What Fazana showed from our side was actually the in vivo ontogeny that she presented was similar to the existing in vitro ontogeny for CYP3A4, whereas Upreti presented an overexpression of 3A4 in some of the early ages that then dropped off to the adult level, which was an interesting observation. They were sort of, I'll come on to how both of these analyses were performed in a couple of slides. In fact, the next slide. It's interesting that the Upreti CYP3A4 ontogeny, showing this overexpression at the early ages going down to adult levels later on. They actually weren't the first to suggest this, because Andrea Eggington, in one of her early papers, I think she used cubic spline function. She actually came to not as extreme, but a similar conclusion. And you can see on the graph here, I put the data points on from Andrea's sort of analysis, and as I say, it's showing this overexpression going down to at younger ages, going down to adult levels later on. So what we were left with then was a bit of a dilemma in a way, that we'd got these two in vivo derived ontogeny functions showing different things. And actually, what we put into Simcyp at the time was profile 1. So from version 15, Fazana's CYP3A4 ontogeny, which, as I say, wasn't that much different from the existing in vitro ontogenies, went into Simcyp. Forgot to say as well that Fazana used IV midazolam as her in vivo probe, which she then deconvoluted back to intrinsic clearance values, and they express those values relative to an adult value to give an ontogeny. She also tried to model for disease as well, but that is actually quite a difficult aspect of deconvoluting in vitro data back to intrinsic clearance values and an ontogeny profile. The paper by Upreti was perhaps slightly scanter on methodology, but they'd used sufentanil, which is an anesthetic agent, and they used weight normalized pediatric sulfentanil PK data to generate their ontogeny. So actually, because we had both these ontogenies from version 20, both of these were put into Simcyp. But then, the question gets asked, it's not an ideal situation having two ontogeny profiles, profile 1 Salem, profile 2 Upreti. What I should just say before we go any further, actually, that we actually did modify the Upreti model in Simcyp when we applied it in Simcyp from version 20. We increased the F birth slightly from 0.1 to 0.15. So this means there's more expressed at birth, and we based that on IV midazolam clearance in neonates from the two studies you can see on screen there. We also set the age cap in equation 2 to 12.5, and that was really to prevent adult values falling below 1. And we removed the C3 term from the Upreti. We've actually just done this because actually it's a fairly redundant term. And what it should be, if you look at equation 2 on screen here, the equation 2 should really just, the exponent should be C2 times age minus age cap. So it's obvious that, having two ontogeny profiles was unsuitable, and, it makes sort of dosing projection difficult and uncertain. And the regulators often wanted to see results from both profiles, and this sort of made life difficult for certainly our consultancy team and any consultancy projects we're involved in. And I remember from this study I did on mefloquine, I had to present for the German regulators both the Salem and Upreti ontogenies. And there was also an interesting paper from the Manchester group, which is the second one on here, looking at DDI prediction, we'd done a few papers on predicting DDI, and actually to predict DDIs in paediatrics, especially when you've got two enzymes there, you need an accurate representation of all the ontogenies to see if there's going to be any difference in your DDI liability with age for that drug. So evaluating the two CYP3A4 ontogeny models, this is a study I did last year, actually. I'm calling this study 1. So basically, I used midazolam data to look at the CYP3A4, the two CYP3A4 ontogenies that were in SimSip. I also went, and I'm not going to cover this, but I also went on to use 1-hydroxymidazolam data to look at some of the UGT, UGT2B4 and 2B7 ontogeny in vivo as well. But I'm not going to present any more on that today. So basically, I had 9 published studies on midazolam, IV PK in paediatrics, and that was term neonates to 17, and 1 study in preterm neonates. And I basically compared the Upreti ontogeny with that of Salem et al. So this is quite a busy slide. So in terms of the results, if we look at the graphs first here, I'm showing basically representative results from 4 studies. And I'm comparing on the left-hand side graphs. So the left-hand side graphs A, C, E, and G are the Salem ontogeny, and the right-hand side graphs B, D, F, and H are the modified Upreti and Wallstrom ontogeny. And these are studies done in sort of different age bands of children. Von Groom is sort of quite young, sort of neonates, early infants. Reed et al. Is some paediatric studies. Some were up to 2 years old, some were older. But you can see in general, if you compare the Salem and Upreti ontogeny models just visually from these graphs, you can see actually Upreti and Wallstrom appears to be doing better for a number of these studies. So what we did across all studies, based on the either the AUC 0 to infinity or the clearance prediction, that's the predicted to observe values, we looked at how the 2 models were performing across the 9 studies we had, and we compared the mean squared error, which is precision, and what we showed there was actually if you compared Salem to Upreti, the Upreti was more precise, and also in terms of mean error, the bias. Again, the Upreti was performing quite a lot better than Salem. So higher precision and less bias for Upreti. The sort of weakness of this study, and actually the reason at the end of the study why I go on to say, we need a broader verification exercise on this with other drug substrates in, was that actually some of the studies here were also used by Salem to generate her ontogeny. So this was a sort of not a major problem because it was only 3 out of the 13 studies used by Salem that were actually crossed over in this study as well. So, as I said, further studies were needed to underpin the generalizability of the optimal CYP3A4 ontogeny for other drugs metabolized by this enzyme. So, what we then went on to do, and really this was as a paediatric team in SimSip, we asked one of our members, Mark Kodashione, to actually do this study. So this is the second study that we did on this theme, really, to compare the Salem and the Upreti models. And this is prediction of pediatric PK for CYP3A4 metabolized drugs, comparing the 2 ontogeny models. And what we hopefully did here was a much more thorough approach compared to that first study that I'd done. So the overall project aim was performance comparison of both CYP3A4 profiles. And in terms of methodology, in terms of literature search, we looked at clinical studies where 3A4 substrates were administered IV. And we focused on compounds where the FM for 3A4 was greater than 80% in the adult population, with a low to medium extraction ratio. We look for low to medium extraction ratio drugs in children. And the final drugs selected were the midazolam, alfentanil, fentanyl, and sildenafil. Now, you could argue that actually in adults, fentanyl is a fairly high extraction drug, but actually for most, especially the early pediatric groups, it's not high extraction. It's sort of a more intermediate extraction compound. So we also put in fairly strict exclusion criteria as well in this analysis. So we avoided any clinical studies used in the development of either profile 1 or 2. So that was either midazolam files, and we didn't use the serpentine actually in this analysis because it failed our sort of inclusion tests. And we tried to avoid data from critically ill children or those having cardiopulmonary bypass or ventilation, or where insufficient PK parameters were reported. And we tried to exclude studies with high numbers of preterm subjects where we couldn't differentiate the preterm from the term neonates. So, as I say, we were left with these 4 substrates with the alfentanil, fentanyl, midazolam, and sildenafil. What this graph really shows you is the studies that we selected and the age range that they're covering. So I think this is sort of, in a sense, interesting in its own right, because you can see, looking at the left-hand side of the figure here, some studies you've got in fairly narrow age bands. So one of the alfentanil studies here, one of the midazolam studies here, a fentanyl study here, and another midazolam study, and another fentanyl study were done in fairly narrow age ranges. But if you look at this midazolam study at the top here, it went from, I think the study was 0.5 years up to something like 16 years. So it's covering a wide age range. And often, within these studies, they really just summarize PK parameters for the whole population. And that sort of makes it quite difficult, really, to use those particular studies for ontogeny type work when it's not being split down into more discrete age bands. So the way that Mark did this, really, he looked at some of the performance, really, of the 2 ontogenies, similar to how I'd done this before. So again, here you're seeing the Salem ontogeny in this set of 3 graphs and the modified Upreti here. And this was for 3 studies in the very youngest age group, where it was 3 of those studies I've just highlighted on the previous graph, where the age band was quite narrow. And you can see here that actually there's a marked improvement for the modified Upreti compared to the Salem ontogeny for these particular studies. What Mark then went on to do is look at some of the older ones as well. So these are sort of, mid-age range studies, sort of 4.5 to 7.7, 2 to 7, and 3 to 10 years. And as you'd expect, by the time you get into these children, you can still see some modest improvement for the modified Upreti model, but it's much less so than in the previous age group, where obviously ontogeny is more important. So what Mark did by sort of splitting down the studies like this, he was able to sort of focus in on specific age ranges where you're likely to see most difference. He presented all the data, including those wide range studies as well. So he split it down into sort of 0 to 2, 2 to 10, and then the broader range studies as well in terms of his analysis. So what he also usefully did was this sort of figure here. Now, what he's done here is sort of summarized all the results. So on here, you can see all 4 3A4 substrates, and you can see the observed AUC to infinity or clearance value, and again the predicted values using the Salem or modified Upreti ontogeny. And then in the final column is looking at the predicted to observe ratios using both of these ontogenies, and he's done like a traffic light system here to make it easier to visualize. So in the Salem ontogeny, you can see more red traffic lights. That's where the predicted to observe value is outside the 0.5 to 2-fold range. And then you've got the amber, if you like, range, which is 0.5 to 2. And then those which are less than 1.5, the 0.67 to 1.5s is showing in green. And you can just see visually here, actually, yeah, the modified Hepatic ontogeny did better in terms of, predictions within that 0.67 to 1.5 fold range. He went on then to look at the results in their entirety, and he looked at the average fold error and average absolute fold error, as well as markers of bias and precision. And this is where he splits it down into the different. He looks at all the data, which is the top line in this table here. So that's all the studies he's looked at, and you can see that actually, in terms of average fold error and absolute average fold error, the Hepatic model is performing better. And especially if you look in the 0 to 2 years old, you can see that there's a marked improvement of the Hepatic model. Less so, it's more marginal in the 2 to 10 years old, and in the overlapping studies as well, you're still seeing some minor improvement using the modified Hepatic model. So in terms of the Hepatic CYP3A4 ontogeny conclusions and future research then, so what we're doing within SimCyP, we're retaining the modified Hepatic profile. Has this performed better than the cell-end profile for the 4 substrates in paediatrics? And as I say, this last study backed up the previous one I've just done in midazolam. One of the problems has been that age bands for certain clinical studies were very wide, and I think that this is something that I would encourage sort of people doing clinical studies, and I do fully understand how difficult some of these studies are to perform in children. They're not easy, but to just try and get the critical mass so they can split the data down into specific age bands would be extremely useful. Got to mention here as well that this analysis was really limited to SimCyP and did not consider other PBPK platforms. Now, you assume that most of the underlying structure of these models is fairly similar, but it basically is up to users of other platforms to do their own verification on this, really. One of the things we want to do is try and expand this to other substrates, which is not particularly easy. And the other thing we need to probably look at more, especially in neonates and in early neonates, is the role of 3A7 in the metabolism of some of the drugs. Now, 3A7 has been shown using recombinant data actually to have a very limited role in midazolam and sildenafil PK. But there was an interesting study recently on metronidazole where they showed actually 3A7 could be important for the metabolism of that drug, especially in the very early ages. Now, I think metronidazole is metabolized by a range of enzymes, and actually the sort of main metabolizer of metronidazole change a lot with age. But they were concluding that for the very young neonates, and especially preterm neonates, 3A7 could have a role. As I say, we did look at this, and we found studies on midazolam and sildenafil for recombinant 3A7, and really the sort of contribution was absolutely minor. And we've actually tried to model some of the data in SimCyP for midazolam, and I think even in the very preterm, I think at most it was only 12% of the metabolism of midazolam was due to CYP3A7. I should have said, sorry, that 3A7, for people who don't know, is the fetal form of 3A4, and there's a change at birth, a big drop-off in CYP3A7 levels, which actually is expressed in very high levels in preterm and early term neonates. That level of 3A7 drops off very quickly, and 3A4 becomes the most important enzyme in the neonatal period. So the conclusion is actually, we may be needing to factor in 3A7 for other compounds, but for these compounds, we did at least consider it. So there's been some other studies that did select the CYP3A4 ontogeny and did conclude actually that it was possibly better than the CELEM ontogeny. And actually, some of this is fed into the reasons why we've done some of this work as well. So they're just highlighted on the screen here. So in terms of just moving on to intestinal 3A4 ontogeny now. So, obviously we'd done this work on hepatic 3A4 ontogeny, concluded that CYP3A4 was okay. So then what I went on to do was this study, which was looking really at the on combining data on the bioavailability of midazolam and PBPK modeling to investigate intestinal CYP3A4 ontogeny. So this has been a long-term interest of mine, really, and I'm just flagging this slide really sort of almost summarising how gut metabolism is handled within the SimCYP simulator. But this was a study which was really part of my PhD more than 20 years ago, and basically I gathered, or through the paediatric gastroenterologist at Sheffield Children's Hospital, I collected I think 105 duodenal and jejunal biopsy samples from really neonates onwards, and I measured using Western blotting CYP3A4 expression, and I also looked at the activity as well in S9 fractions from the enterocytes that I'd managed to collect as part of these biopsies. And you can see on this graph here, what I was showing was a definite ontogeny for both expression, which is this top graph, and also activity. I should say it's in vitro activity of the enzymes as well. I didn't actually collect the fetal samples. They were a gift to me from one of the London hospitals, but even in the fetal ones, I did actually manage to detect some very low levels of enzyme using the Western blotting. So that's why I'm almost revisiting this, 20 odd years later, revisiting intestinal 3A4 ontogeny. So this is just summarising how we deal with it in SimCYP. We in the adult model, we look at the overall intestinal expression of 3A4. And in the paediatrics, you have to take into account the relative gut surface area and also the ontogeny here. And, via an intrinsic clearance and one of two models, either a first order model using the Q-gut model, which is a the Q-gut sort of model is similar to the well-stirred liver model, but what Q-gut is, it's not just a blood flow, it's a permeability and blood flow term as well. So it's permeability of your drug. You can often measure that through CACO2 cells, and also the villous blood flow, which is around 6% of cardiac output. So obviously the villous blood flow changes with age as well. And from these, either the first order model, which includes that Q-gut, or the ADAM model, which has gone beyond Q-gut actually. So this I'll come on to later, but it's a segmented model. So in the ADAM model, you've got the intestinal expression of your enzyme and your blood flows, etc., all in different segments of the GI tract, representing duodenum, sections of the jejunum and ileum as well. So it's a more mechanistic model. So from all this, from these two models, you can calculate the bioavailability. And I'll come on to this graph here in a minute. So what the aim was to really compare and rank published intestinal 3A4 ontogeny models by assessing midazolam bioavailability. So we conducted a detailed literature search on midazolam oral bioavailability in adults and children, conducted another literature search on intestinal CYP3A4 ontogeny in children, and we basically tested the different models that we came up with. And these are shown on the next slide to best describe changes in oral midazolam bioavailability as a function of age. And what we did, we used two compound files, one assuming all CYP3A4. So we modified the SimCYP midazolam file, which also includes 3A5. We used the retrograde model to actually just assume all the metabolism was by 3A4. And you'll see why I did that in a moment. And we also went on to look at the likely expression in preterm neonates as well, but not really going to say too much about that. So these are the different ontogeny models we came up with, and you can see that they're massively different. this study here is Chen et al. 2015. They looked at mRNA, and quite interestingly, actually, they're not the only ones to show this, because May Thackeray, who was working for Evelyn Jacques-Agian's group in Paris, has also shown a similar mRNA ontogeny for intestinal 3A4. But there's a mismatch between, it seems to be a mismatch between mRNA data and protein data, which are these bottom graphs here. So this grey line here is Kiss et al. So this Saskia de Wilt's group, they used a proteomic approach, as did the Gulen group, which was University of Birmingham at the time. And these are more recent studies, Kiss et al. And Gulen et al. And they used a proteomic approach, and they also looked at an array of other CYP, UGT, and transporters between these studies. The interesting thing about this study by Kiss et al. As well, they looked in different segments of the GI tract. So what they showed in this paper, Kiss et al., was that they showed a definite ontogeny in the ileum, but in the duodenum, they more or less showed no ontogeny. So that's why we've included this no ontogeny model here. This black line is my ontogeny that I generated all those years ago. So, oh, and I should say as well, we put in the modified Upreti model mainly because a recent study had sort of used that as both their hepatic and intestinal 3A4 ontogeny model. But, you know, there's no sort of direct evidence that you see this profile in the intestine. So as part of sort of this as well, we wanted to sort of get a good handle on the bioavailability of midazolam in adults. And this is really a summary. There's lots of data, 36 studies I found, range of values 0.096 to 0.68. And actually, if you look at this data, only 6 of them were outside of the range 0.2 to 0.45. So the weighted mean was 0.3 in terms of bioavailability of midazolam in adults, with an overall standard deviation of 0.14. Interesting data in its own right, really, in that only 2 studies really considered the effect of 3A5 genotype. Karash et al. 2007 gave a PM value of 0.26 and an EM value of 0.22. While Kvitny et al. Gave a pure PM value of 0.46. Now, our simulated values for PM were 0.34 and EM were 0.17. So actually, we showed a significant difference between the two, whereas that original study of Koresh didn't. So that was interesting. And, you know, I probably would say that it probably needs looking at again in the adults in terms of a bigger cohort of EMs and PMs compared directly. So this was the bioavailability data I could find in Paediatrics, and I'm expressing this on a log scale. This is post-menstrual age in weeks, and this is the bioavailability here. And we can, you know, I only found basically 6 studies. Now, some had more than one value in different age bands, so I was able to include those. So what you really can see is actually this line here is demarcating preterm neonates from term subjects. So what you're showing here is actually, yeah, you're seeing some sort of change in bioavailability, and especially in these young ages, bioavailability seemed to be higher. By the time you get into around a year here, you know, you're not really seeing that much change in bioavailability. I should say that this point here, this like horizontal striped circle is the adult data. So, you know, I've tried to represent the size of the study by the size of the shape here as well. Obviously, I can't go to the full size of the adult study, it just obscure the graph, but it gives you an idea. So most of these studies were done, most but not all were done in a standard crossover way for bioavailability. So 1 or 2 were done in a non-standard way using PBPK, or a non-crossover type study. So that these results here from Payne were a cohort of 1 population given IV and 1 population given oral. So in that sense, it's not a true bioavailability study. So what I went on to do was really compare using the first order model in SimCyp And in this case, assuming no 3A5, so I'm assuming all midazolam metabolism is by CYP3A4. And what I'm showing here is the different ontogeny models that we put into SimSip. And I'm superimposing the grey dots here are really the SimSip simulated individuals, and then the black line is really like a moving average line. So what I did to generate the black line was to set all the variability to 0 in SimSip. So that's how that was done. And then I'm superimposing on the clinical data, which I showed in the previous slide. So it's more difficult to sort of discern in a way than the hepatic CYP3A4 ontogeny. But, just eyeballing it, my model doesn't seem to do too bad. Kiss et al. Seems to miss out here. The Gulen model, I should have said about this as well, that they didn't manage to generate much data. They didn't generate any data below 11 months in terms of their clinical samples. So, it was a bit of an extrapolation back to, birth with that, which, is not ideal. It would have been better if they could have generated some data in the 0 to 11 months. It highlights, again, the difficulty of doing some of these clinical studies when, if you don't get the samples coming through, there's nothing really you can do about it. Obviously, the Chen et al. One, which was based on mRNA data, doesn't do very well. So between these, the Johnson one, the no ontogeny, and the Upretti and Wallstrom seem to do best. And I've got to remind you that's not an intestinal ontogeny that's been discovered. This is just assuming the intestinal and hepatic ontogeny are the same. I went on to do some more results as well using the SimSip midazolam file, which obviously incorporates CYP3A5. On these graphs, that's difficult to see. Similar to before, really, the solid black line is the average from the EM and PM. The dotted dash line, which is above the solid black line, is a moving average for the PM, and the dotted line underneath is the moving average for the EM. So obviously, you're putting more uncertainty into the model, and it again, you can say that, well, maybe Johnson, no ontogeny, and Upretti are doing okay. So what I did at the end was I compared average fold error and absolute average fold errors for the two models. As I said, the results were more difficult to sort of discern, but there was a general pattern. So in terms of using just CYP3A4 midazolam model, in terms of bias, no ontogeny was less biased than Upretti, which was more or less the same as my model, and it was better than Gulen, Chen, and Kiss. And in terms of precision for this model, my model performed better than Upretti, then no ontogeny, Gulen, Kiss, and Chen. And it was similar for when we had the other model, but some of them were sort of more uncertain in terms of precision. Mine was equal to Upretti in this case. And in terms of bias, the no ontogeny performed best, then Upretti, then mine. So, coming to an absolute conclusion is more difficult, but what you can definitely say is there's some sort of ontogeny for intestinal 3A4. And actually, overall, because my model didn't do badly, and as I say, it's more tenuous than the sort of hepatic ontogeny. What I did in the end was thought, well, actually, my CYP3 intestinal 3A4 ontogeny is not doing bad. There's no great reason to change it based on this work. So what I'm showing in this graph actually is I used my CYP3A4 ontogeny, the default one in SimSip, and I thought, well, I'm going to compare the first order and the Adam model, and it's using the same ontogeny model in each. And as you'd expect, the two different approaches, it gives you quite a lot of variability, more so this time, because this is based on a slightly different approach to the previous graphs I've shown you. So this is looking at AUC ratio as a marker of bioavailability. It was the same dose IV and oral given to the subjects. So you see a lot of variability, but actually when you look at the two models in terms of the F value predicted by Adam and the F value predicted by first order, there's a good correlation, 0.93. So, that was interesting. And what the Adam model will allow us to do, and it's sort of something I've sort of mentioned already from the Kiss et al. Data, is that if there is a different ontogeny for different segments of the GI tract, then via the Adam model, we could potentially put those into the Simcyp Paediatric model. So I just wanted to flag this study. This was a very recent study, really published while I was doing that intestinal CYP3A4 ontogeny evaluation paper. And this is from Tom Deval, who's working with Patrick Augustin's group, which is Leuven, I think, in Belgium. And they presented again some proteomic data on intestinal an array of intestinal enzymes and transporters. Actually, if you go into the paper, you can see the range they've done. And what I'm showing on this bottom left-hand graph here is, I think that they didn't have that many subjects. I think they got 11 neonates and 7 pediatric subjects. And the colours of the different symbols here are really it's really split into duodenal are blue, jejunum's red, ileum's green, cecum's purple, and transverse colon's orange. And the solid symbols, the neonates are closed symbols, and the open symbols are pediatric. So you can see in the duodenum here, even though they're individual subjects, there seems to be a sort of definite ontogeny for CYP3A4 in the duodenum. But as we've got so few samples in some of the other areas, and in the ileum, there doesn't look like there's an ontogeny at all. So I thought it was interesting data. It's limited data. And one of the other problems, and I did write to the group and ask them whether they did have any adult data, but nothing came back from them. So what I decided to do actually was use the two oldest subjects as representative subjects, because it seems to, if you look at the graph here, even though it's condensed, it seems to be coming to a plateau here. So I used the top 2 points here as if they were adults, and then I expressed the others as a fraction of adult. And what I'm doing here is I'm comparing my model, which is the blue ontogeny model, with their individual data. And one of the things that they may be suggesting, although this data is limited, and it's something that would help the performance of the intestinal 3A4 ontogeny we have, is if we put the F birth to lower and make it develop slightly faster, it might give us more of an optimal result. So in terms of intestinal 3A4, conclusions and future research, there's a definite ontogeny for intestinal 3A4. But the optimal models, as I say, it's quite difficult to discern, especially after considering 3A5 and general variability. So my model with a lower F birth and a quicker onset may be closest to recovering the observed data. That may be the optimal model based on currently available data. There appears, as I've already said, to be little age-related change in midazolam bioavailability beyond 1 year. The caveat is it needs confirming with other substrates potentially, but then what substrates? That was the big question, and often, I could look at other 3A4 bioavailability studies in pediatrics, but they tend to be few and far between. So, there's perhaps a hole in the clinical data there to do that, especially sort of lack of clinical data in the early age groups also is an issue, which comes back to that slide I showed you earlier on for the hepatic CYP3A4 ontogeny, where there were a few studies in very discrete age bands, which is a limitation. So overall conclusions and future research. So modified hepatic CYP3A4 ontogeny of Upreti and intestinal ontogeny of Johnson will be the default profiles from Simcyp version 14. What we will do is retain the Salem hepatic 3A4 ontogeny, but it won't be the default one. So it will still be available for back compatibility in terms of old workspaces or whether somebody wants to use it. Other PBPK platforms, users should perform their own verification. As I've already said, acknowledging also the difficulties in performing paediatric clinical studies, but where possible to try and encourage people to report in discrete age bands and even try and expand the range of data they've collected to allow for this, really. And sort of future research, I'm not really going to say much about, but we do plan to investigate optimal ontogeny of an array of other enzymes as part of the paediatric team, including CYP2C9, 2C19, and 2D6. As I say, this is a team effort involving the whole sort of paediatric team at CYP3A4, really. So that is the conclusion of my talk, really. So I'm happy to take any questions if anyone has them.
Hey, Trevor, again, nice talk. There is a question in the chat, so I'll go ahead and read that to you. And I think I see maybe even a couple of raised hands. So Trevor, this comment and question comes from Mayor Lattimore. First of all, I want to pass on his compliments. He says, nice work. And the question is there a correlation between the presented hepatic CYP3A4 ontogeny obtained from clinical probe substrate PK data and the LCMS-MS proteomic data? Considering that obtaining probe substrate PK data for other enzymes and transporters can be challenging compared to protein abundance data. So any insight there would be appreciated. So just relative difficulty versus applicability in clinical setting of
sorry, can you just repeat that, the sort of main part of the question, Jean? I didn't
Sure. What's the correlation between the in vivo ontogeny versus the proteomic ontogeny? Like, is there good correlation?
Generally not, no. So looking at the hepatic ontogeny suggested by UPRETI from the proteomic data, in general, the protein data for CYP ontogeny doesn't go above 1. It suggests some sort of sigmoidal ontogeny going up to 1, but doesn't suggest going over 1. So it's the clinical data rather than the protein data that suggests the profile may go above 1 and come back down to the adult level. So there may be some sort of, in terms of protein expression and activity, you'd expect a strong correlation, really. Because actually that is what I showed for intestinal, and other people have shown it as well for hepatic, but I certainly showed that for intestinal 3A4, that the activity and expression were sort of linked, really. So I'm surprised. It's surprising, and it's a difficult thing to sort of explain. Why are we seeing this over expression generated by activity data, but we're not seeing it by proteomic data? And that's always been a slight concern. I've got to be honest. I think that's a pertinent question that's been asked, and to sort of try and explain it in your own mind is quite difficult. But that's what that's what people have shown.
There's another question that just came in. So from Ming also says, thank you. What are the most recent experiences from CYP consultancy projects with regard to regulatory acceptance, a pediatric dose projection, and DDI risk assessment of 3A4 substrates?
I think that they've got an increasing number of sort of projects that have gone through. I was speaking to them a few weeks ago. I think often they're still getting asked to do both profiles. And this is the reason for doing this work. And actually, the regulators wanted to see this work, and they wanted, from the paper that Mark did, the regulators have asked for all the data. And actually, for a recent submission, they submitted all that data alongside their work that they'd done, where they'd used the UPRETI ontogeny to generate dose information in children. So this is what the transition we want, really. We want to move away from doing both the CELM and UPRETI ontogeny to doing the UPRETI ontogeny. But as I say, the last time when that was submitted by the consultancy team, the regulators wanted to see all the data from our study to show that how we'd verified UPRETI was better. So I don't know if that answers the question particularly, but yes, that we're moving away from both ontogenies, but as part of this, the regulators are wanting, verification data, and we were able to show them that from sending them the actual files that Mark had generated in Simcyp and that we'd used to generate the paper.
Yeah. I think a second part of the question was with regard to DDI risk assessment of 3A4 substrates. Don't know if you can speak to that and the different ontogenies and the regulatory acceptance of DDI risk assessment.
Yeah, I mean, it's similar really to almost dose projection. And, the University of Manchester paper highlighted that I presented earlier, that if we're looking at DDIs and it's a DDI where there's two enzymes involved or something like that, then it's important to have, a good handle on the actual ontogeny. So I think what our consultancy people have done in the past is they've done DDI assessment using both ontogenies, but we want to now move down to this Hepatic ontogeny. I've got to say, it's only really important if it's a compound that's purely metabolized by 3A4. It's not that important because whatever ontogeny you put in there, if it's purely 3A4, you're going to see a significant interaction anyway. It's when you've got two enzymes that may change in importance with age. I think the DDI question of ontogeny becomes more important, really.
Thanks. Any other questions? Also, feel free to unmute and ask the question.
If nobody have got a question, I have one question for Trevor. In the operative table that for the result, when you were showing that, it was one case that in operative it was, I think, over predicted. Do you know what was the reason for that? It was 2.4 fold.
Let me see if I know what you mean, Masood. Yeah, there was one. Let me.
Yeah, that one.
This one.
Yeah.
The Killian study.
Yeah.
That was a study done in quite early neonates. I think it was 0 to 0.01 year. So these are like 0 to a week old. So I think, in terms of one of the difficult things is what is the true value at birth? And, even a small change in that could influence the result. I think the original Selem ontogeny had a lower fraction at birth, so it may be that actually, we need to change the F birth in the Hepatic model slightly to sort of recover that. But yeah, it was one study in very early neonates, 0 to a week old, really. So I think there's still uncertainty there in terms of F birth values.
And the last one, the gauntlet study is in this one is the other way around for both ontogenies.
That one is again quite early on. That's 0.027 to 0.16. So that's like birth to 2 months.
Okay.
So yeah, it was a couple of studies that were in sort of neonates really, where the predictions weren't great. It can be for other reasons as well, Masood, in terms of how the clinical studies were done and how well we were able to replicate them in Simcyp. And this is I'm doing some work currently on CYP2C19 ontogeny, and some of the studies they give children different doses, and then you just in terms of doing the simulation, you're left giving them an average dose, and sometimes that gives you misleading values.
Okay, thank you very much.
I think there's two parts of that. One, we might be able to optimize the F birth a bit more, and two, I think sometimes on the clinical side, it's difficult to replicate it because of study design, etc.
Thank you.
Okay.
Okay. Well, if there's no other questions, currently there, Trevor, if you wouldn't mind going to the last slide, please.
Sorry, we've got to.
No, it's okay. just currently there is not another webinar scheduled, but please, when that's available, registration link is there for your information. So can go ahead and close out the meeting, but we look forward to seeing you at the next webinar. Thank you, everyone.
Thank you.